again welcome everyone to this Thursday webinar. So my name is Mariangela Pellegrini. I'm the project manager for the ERN-EuroBloodNet, so European Reference Network on Rare Hematological Disease. And I'm very honored to welcome all of you on behalf of all the ERN to this session. Before starting, I would like to share with you some home rules and technical information. So as you can see, this webinar is recorded because we implemented all the video in our e-learning environment. So if you don't feel comfortable with that, please put your camera off or change the name in the platform or notify to me and I will take care of it. And also kindly notice that your microphone are muted right now. If during the presentation you have questions, you can write them in the chat, or at the end of the presentation, of course, we will have the Q&A session when you can both raise your hand and I can pass the floor, or write a question in the chat and we will tackle all your feedback, comment, question, curiosity that could come. So coming to the program named Thursday Webinar. So this is a program of the ERN that aims to promote the interest on very innovative topics among health professionals, focusing the cutting-edge advances in the field of the rare hematological disease, which is the scope of our ERN. So with this objective, today we are going to assist to a lecture of Professor Antonis Kattamis, and we will talk about gene therapy in hemoglobinopathies. Just for quickly recall, the speaker of today is international expertise. So Professor Kattamis is professor of pediatric hematology oncology at the National and Kapodistrian University of Athens. He is the head of the division of hematology and oncology of pediatric at the Hagia Sophia Children's Hospital in Athens. And the thalassemia and oncology unit of this division has been recognized as National Center of Excellence for Rare Blood Disease, Pediatric Cancer, and Cancer Predisposition Syndrome, and is also, of course, full member of the ERN-EuroBloodNet PETCAN and Genturis. So please, Professor Kattamis, the floor is yours. Thank you very much.
Thank you, Mariangela, and thank you all for being here today. And I'm going to give my lecture on gene therapy in hemoglobinopathies. These are my disclosures. And I will just start with some of the announcements that gave a lot of enthusiasm in the field of hemoglobinopathies. It's only on the last 3-4 years that we have seen some of the gene therapy products to be available for commercial use. Starting for Zilteglo from the European Medical Medicine Agency in 2019, and then from the FDA for thalassemia, and just recently for sickle cell disease. And why this is very important? Because we do know that despite the treatment that has been developed over the last year, and we have seen a dramatic improvement in the quality of life and the survival for patients, it's still, both for sickle cell disease and thalassemia, the survival remains suboptimal. And it's only around 80% of the patients with thalassemia, transfusion-dependent thalassemia, that will be able to celebrate their 50th birth date. And something similar is going on around 50% of the patients with sickle cell disease. They will be able to celebrate their 48th birth date. So although we have seen a dramatic improvement, we are still lagging behind in compared to normal population. So there is need for finding some other ways to treat our patients, because we do know the conservative treatment with blood transfusion and drugs like hydroxyurea has a very good effect, but has also many limitations. On the other hand, the main curative therapies with hematopoietic stem cell transplantation is only limited for patients that they are very young at age, less than 14 or in the adolescent years, or patients that do have a patient need to have a complete match donor in order to proceed with bone marrow transplantation and be cured from either thalassemia or sickle cell disease. Therefore, gene therapy gives a new hope for patients with thalassemia and sickle cell disease to being cured. And what is gene therapy? We have two types of gene therapies. The one that we call gene addition, when we insert to the genome of the patient, a new gene, a normal gene, and we wait for the new gene to produce the protein that it's supposed to produce. And then we have gene editing, when we try with some gene editing machinery to alter the function and the expression of the gene, either by disrupting or deleting, or by correcting or inserting a new gene. So we have two different ways of approaching the gene therapy field. The truth is that both approaches have many similarities in the way we do it, we perform it with hematopoietic stem cell transplantation. For gene therapy, we need to collect on the first step, we need to collect autologous hematopoietic stem cells. Then we transfer it to the manufacturing location, where the cells are manipulated, and they either undergo a gene editing process or a gene addition process. Then the manufactured product is returned to the patient, who in the meantime has undergone a myeloablative conditioning regimen in order to destroy the bone marrow, in order to make space for the new infused cells to develop. This myeloablative conditioning regimen is very similar to what was used for the allogeneic hematopoietic stem cell transplantation. If we look on the list of the clinical trials that they are focusing on gene therapy for hemoglobinopathies, and these are only the trials that are currently active, either recruiting or ready to recruit patients, you can see that the list is very big. On the top part of the list is the gene addition trials, with the most prominent and the more advanced data are from the trials by Bluebird on the beta-glucan autotem cell product. This, that was initially reported in 2016, and is the first gene therapy that actually was approved for commercial use in 2019 by EMA. I use a lentiviral vector, which carry a gene cassette, in which we have the beta-globin gene, along with all the elements that they are needed for the gene to be expressed. This globin, the beta-globin, has a polymorphism at the amino acid at place 87, which has two functions. First of all, has some anti-sickling properties, because it actually affects the contact point of the hemoglobin, and also it can give a very good indication. You can use that for evaluating the production of the new gene, as it can be very easily separated on common HPLC method. If we look a little bit at the program of the transfusion-dependent thalassemia, you can see that there was phases with initially non-beta 0, beta 0 genotypes, and then with beta 0, beta 0 genotypes. And in the most recent Congress of the American Society of Hematology, the results of these three phases were combined and presented. You can see that overall they had 53 patients that they were transplanted. Of them at the phase 1 and 2, the majority of them were non-beta 0, beta 0, as was also in phase 3, but there was also some beta 0 patient. The mean age of the patient was fairly young, 20 in the phase 1 and 2, and 13 in phase 3. And also you can notice that there was some patients that had already undergone splenectomy, 9 in phase 1 and 2, and 17% in phase 3. And if we go directly to the results, between phase 1 and 2 and phase 3, there was a change in the manufacturing process, and the product was more potent, and was the process was more efficacious. So you can see that out of the 22 patients that participated in the phase 1 and 2 trials, 15 remained transfusion-free with a median follow-up of 92 months and the longest follow-up of 9 years. Within the phase 3 trial, there were 41 patients, and more than 90%, 37 patients were transfusion-free, with a median follow-up of 50 months and a more than 5 years follow-up of 10, and we had 10 patients that had a very long follow-up time, more than 5 years. You can see there was not big difference in regard to age, and there were patients that they are very young, less than 12, adolescents, and older patients. The studies have shown that the peripheral blood vector copy number remains pretty stable. That's on the right. Phase 3 is a little bit better. It's around 1.5 of vector copies for diploid genome. And for phase 1 and 2, it was around 0.7. But the most important, the level of hemoglobins remained pretty stable throughout the observation period. The red line represents the phase 3. You can see that the median hemoglobin was around 12, and that was persistent throughout the follow-up period, with the majority of the hemoglobin to be produced was from the new inserted gene, as you can see from this graph. The blue line represents the phase 1 and 2. Here the hemoglobin levels remain a little bit lower, around 10, and the hemoglobin that was produced by the vector that was introduced was around 6. In the last American Society of Hematology meeting, they also presented the results on the follow-up on the iron overload. At month 48, there were 30 patients that have undergone liver iron concentration measurement, and of them, 21 had a liver iron concentration less than 5, and of them, only 5 required chelation. And out of the 9 that had a LIC more than 5, on the follow-up evaluation, 4 actually stopped iron chelation therapy, and only 5 remain on chelation therapy. So, the fact is that what we see is that overall the patients are responding very well, and they are able to control their iron overload, and sometimes they are able to decrease also the iron chelation therapy. Toxicity, majority of the adverse event occur around the time of the transplantation, and they are related to the conditioning regimen. There were 5 patients, 8% that developed VOD. There was no significant problems. There was no malignancies, no cases of insertional oncogenesis or vector-derived replication or clonal predominance that was observed on the long follow-up of these patients. What about sickle cell disease? Along to the thalassemia studies, studies on sickle cell disease also were developed with this same product. The product now was commercialized as of the early December of 2023, and the name of the product for sickle cell use is Lyfgenia. This approval from the FDA was based on the collected results of the patient. There was again 5 phase 1 and 2 and phase 3. There were 59 patients that were mobilized. Of them, 4 at the time of reporting the data were still in the process of low-vol cell infusion of the manufactured cells, and 8 actually discontinued prior to the infusion. So there were overall 47 patients that were transplanted, and the efficacy and safety pool was on these 47 patients. And the demographic shows that the age was around, the median age was around 23, with a range between 12 and 38. The majority of the patients were actually older than 18 years old, but there was 10 patients that were in the adolescent years. The majority of the patients, 98%, were homozygous hemoglobin S. You can see that there was a number for mobilization cycles was a mean of 2 between 1 and 4, and the estimated average and the total CD34 cell dose was 6.5 million cells per kilogram. We'll go straight to the results. The primary endpoint was complete resolution of all vaso-occlusive events at the period between 6 and 18 months, and you can see 30% out of the 34 available patients who were actually achieved the primary endpoint, and that means an efficacy of 82%. All of the adolescent patients achieved the primary efficacy point, and out of the other 4 patients that still had some VOEs, was a significant decrease in the number of VOEs compared to the 24 months prior to the infusion of the cells. Moreover, the patients were able to maintain a very good hemoglobin level without any transfusions. You can see the mean hemoglobin level was between 11 and 12, and around between 45 and 50% was hemoglobin that was produced by the introduced beta globin. All of them had a durable response, and there was no stroke, even patients that had a history of stroke. There was a significant decrease in all the markers for hemolysis, being indirect bilirubin, reticulocyte count, or LDH, and there was a very good improvement in the patient-reported outcomes. You can see that for pain intensity, 57% of the patients had a clinical meaningful improvement at 36 months, 64% for pain interference, and 64% for fatigue. What about the safety profile? Again, the majority of the safety concerns and adverse events were related to the busulfan conditioning. There was no VOD. There was no vector-related complication or incisional mutagenesis. There was 1 death due to cardiopulmonary disease, which was a problem that pre-existed the transplantation. There were 2 subjects that developed acute myeloid leukemia and died, and that was in group A. And there was one subject that developed a myelodysplastic syndrome, which seems to be transient. And a further report on this case was presented at a recent American Society of Hematology meeting. That was a young man of 60-year-old with homozygous hemoglobin S disease, who also was homozygous for alpha thalassemia, the one alpha deletion from both alleles. This patient developed a picture that looked very much to myelodysplasia with a lot of erythroid-restricted dysplastic features. And there was a clonality that was identified, a very small frequency, 1.4%, which this single variant was of unknown significance. There was no driver mutation or aneuploidy associated with MDS, with different IFO methods evaluated. And the conclusion is that this is more probably due to erythroid-restricted dysplasia consistent with stress dyserythropoiesis, which has been stable for the 2 years that the patient has been followed for this problem. There is an indication that this may have been caused by a alpha to beta globin imbalance due to the pre-existing alpha thalassemia minor that this patient had. So if we're going back to the big list of the trials that are currently ongoing, we go to the middle of the table. There's a trial that is using gene addition, but not for beta globin gene, but for something else that we'll discuss in the next couple of slides. If you remember, you do remember that the hemoglobin switching is happening short after the fetal life, and beta globin is starting to be produced at the last trimester of fetal life, and then it increased dramatically postnatally. On the other hand, gamma globin production is starting to decrease in the 3rd trimester, and then very quickly decrease postnatally to go back to very low levels during their adult life. This hemoglobin switching is orchestrated by different genes, with the main one being BCL11A, which is expressed postnatally, and the expression of BCL11A, it's actually induce a suppression of the expression of the gamma globin, and thus express the production of the fetal hemoglobin in adult life. And this lentiviral vector encodes a microRNA, adapted short harpin RNA, which interferes with the expression of the BCL11A. So in some way, it silenced post-transcriptionally BCL11A. And the first results were presented a year ago in 2022. There were 12 patients, and you can see they had a very good results with decreasing, achieving the endpoint of decreased vaso-occlusive events after the infusion of the drug. In further reports, they saw that the vector copies expressed fairly stably throughout the observation period, and there is a production of fetal hemoglobin in around between 20 to 30% in a long-term follow-up. And in a recent study, they demonstrated that this hemoglobin F levels corresponds to a high number of fetal cells. This is the percent of fetal cells that have at least 10 picograms of fetal hemoglobin, and it's believed this is a number that will consist, will render the red blood cells fairly resistant to polymerization of hemoglobin S, and these results are very similar, even better, to what is observed with hydroxyurea high responders patients. But in order to affect the BCL11A expression, There have been some different gene editing mechanism and trials. The different gene editing machinery that we have available is homologous, among others, is homologous recombination, transcription activator-like effector nucleases, zinc finger nucleases, and CRISPR-Cas9. CRISPR-Cas9 has been shown to be the most potent method for gene editing. And one of the reasons is it has been fairly simple to set up an experiment. It only takes some days to set up a CRISPR-Cas9 experiment compared to weeks or months with the other machineries that have been used. And CRISPR-Cas9 was first described as a natural defense mechanism in Escherichia coli. It induced protection of bacteriophages and plasmids, and it consists of nuclease Cas9 and two non-coding RNAs, the CRISPR RNA and the TRAC-RNA. And from the description to the understanding what's going on, it has been a very long but also very short period that became from the description to be used on a clinical practice. This is very interesting and very impressive how quickly they have moved from the description as a gene editing tool to be used in clinical practice. Just to remind you that Emmanuel Charpentier and Duda received the Nobel Prize because of their work on developing CRISPR-Cas9 as a gene editing tool. And the first product in the more advanced studies using this technique is the Exacel product, which is a non-viral ex vivo CRISPR-Cas9 mediated editing of BCL11A. It targets a critical transcription factor binding site, the GAT1, at an erythroid lineage specific enhancer region. The repair is by double-strand breaks by non-homologous end joining, which results in DNA insertion and deletion, which actually disrupts the GATA1 binding and lowers the BCL1A transcription, and this reduces overall the expression of the BCL11A. The first patients were reported in the New England Journal of Medicine 2-3 years ago, and I will present directly the results from the two studies. The FAL 1111 was presented at ASH 2023. The full analysis sets included 52 patients, and the primary efficacy set 35 patients. You can see the younger patients were 12, 34% were from the full analysis set were the adolescent period, and 65% were higher than that. And there was also beta zero patients, but also non-beta zero patients involved. The number of mobilization cycles were overall 1, but it ranged between 1 and 4. There was a mean median cell dose of 36 million per kilogram of CD34 manufactured cells that they were delivered. The primary endpoint was transfusion independence at month 12 after the last transfusion that was used during the peri-transplantation period was achieved in the majority of the patients. As you can see from this graph, 32 out of the 34 or 35 patients achieved transfusion independence. And you can see that this primary efficacy set was very similar between older patients and adolescent patients. The total hemoglobin levels remain at very high, above 10, and this remained throughout the observation period. and there was no difference between adults and adolescent patients. Fetal hemoglobin comprised the majority of the hemoglobin in these patients, and you can see that the peripheral and the bone marrow blood allele editing remained stable throughout the observation period. As for the safety profile, there was the adverse event was mainly related to the conditioning regimen, and they occurred around the transplantation period. There was no significant delays. There was no death, no malignancies, and as you can see, it was the majority of the adverse event occurred around the transplantation period. And we'll go directly to the sickle cell disease trial that was also reported by Professor Ragul at the American Society of Hematology a couple of months ago. The full analysis set contained 44 patients with a mean age of 21. 12 of the patients were adolescents, 32 were older, and the majority of the patients had homozygous beta S. They were homozygous for beta S. And the number of mobilization cycles, 2, and range between 1 and 6. 4.7 million CD34 cells per kilogram were infused, and the results are shown here. The main primary endpoint was the cessation of veno-occlusive crisis, and you can see that the majority of the patients, 28 out of the 29 patients, remain VOC-free. There was one patient that had a couple of VOCs around a parvovirus B19 infection, and only one patient had some VOC events, and this is a patient with chronic pain, and but the VOC and even the VOC that he or she suffered were milder than from the baseline. Also, what we see, there was no difference between adults and adolescents. The fetal hemoglobin, it was around 40%, and that remained consistent and stable throughout the follow-up observation period. The majority of the F cells were very close to more than 90% were F cells. There was a durable BCL11A editing in the peripheral blood and the bone marrow, and there was significant decrease in the hemolysis markers, both for haptoglobin count and LDH. What about the safety? There was no adverse events. Well, actually, the adverse events were more related to the myeloablative regimen. There was one death due to COVID, but there was no other severe adverse events with chronic complications. There was no malignancies observed. And as you can see again from the graph, the majority of the adverse event and the severe adverse event occurred around the transplantation period. In conclusion, these two studies have shown that the exa-cel can provide a one-time functional cure for patients with TDT and sickle cell disease. The safety profile is consistent with busulfan myeloablative conditioning regimen. And there was no significant concern about mutagenesis on the long-term observation period. Similar to that, there was a presentation in both in EHA in 2023 and in US in 2023, from a group in China that developed a very similar technique, and they presented 5 patients. And you can see initially they presented 5 patients, but then in ASH 2023, they had also some new patients that presented, and you can see there was a very good response, again, with production of hemoglobin F, and the patient remains transfusion independent for as long as 33 months. This is a very similar technique that they were described by XSL. And there was also another group, again from China, that presented in the EHA and updated the results in us, that they use CRISPR-Cas9 to target the PCL, the Kata1 binding site of that lies in the promoter region of the two gamma globin chains. And by doing that, they were able to show a significant increase in the fetal hemoglobin. They presented 8 patients and, 6 patients, and all of them remain transfusion independent with a very good hemoglobin levels above 10, all of it comprised by fetal hemoglobin. Finally, there was another attempt using a similar but a different gene editing machinery with the AsCas12a. Again, try to edit the hemoglobin G1 and hemoglobin G2 promoters, the gamma globin promoters. This was presented in EHA. They had 4 patients that they, as you can see, they were able to transplant with this technique. The gene edition was very powerful and was able to induce hemoglobin production. As you can see, the first patient had a hemoglobin around 16. Half of it, 43%, was hemoglobin F. The second patient had a hemoglobin of 12.5. The other 2 patients were a little bit early to give definite conclusions. And you can see that the hemoglobin expression was pancellular. So we have these 2 different techniques, the gene addition and the gene editing. There is some concerns. For the gene addition, we need to make sure that there is a continuous expression that is required. There's always fear and threat for insertional mutagenesis. The ratio between alpha and beta globin still may remains unbalanced, as we have seen with the case of a patient with sickle cell disease that developed erythroid dysplasia. Ineffective erythropoiesis still remains, and the cost of the vector still remains pretty high. For the gene editing, the results probably are more permanent. We don't need a vector, but we use a mechanical way to do the gene editing. In the most of the case, we use electroporation, who does not need the vector, but on the other hand, may have a lower efficacy and decrease cell viability. Insertional mutagenesis is less even when vector is used. There is this concern about off-target activity. And what we have seen sometimes, there is a concern about a normal gene function and genome instability, which may be induced by a large chromosomal rearrangement, and who may accumulate. And there's also, we might develop some clones that they have p53 activation, and this has been shown in some of the experiments, which we don't know what's going to happen in the long run. And on the other hand, the low ratio of homologous recombination compared to non-homologous end joining, we might have a disruption of the hemoglobin B gene production, who might develop a thalassaemic phenotype. And we need not forget that the fetal hemoglobin, especially for thalassemia, is inferior than in delivering of oxygen to hemoglobin A. So we have discussed a little bit about the homology-directed repair, and this is the focus of some of the newer techniques like the base editing. This now is on preclinical development, and it's something that we would like to see in the future, as we'll discuss in the last slides. So in the summary, and this is the summary from the last presentation from the American Society of Hematology meeting, the FDA announced the first day of the meeting in December 8th that they approved 2 milestones treatment for sickle cell disease, the gene addition product by Bluebird Bio and the gene editing product by Vertex CRISPR. The latter was the first approved treatment using this technique. And we need to all commit, both scientists but also pharmaceutical companies, to commit for a long-term safety evaluation. So we need to have long-term data in order to make sure that this procedure is safe on a long-term basis. And what has been discussed, both drugs have a very significant cost. Leukemia, which is the gene addition product, the cost in the United States is estimated around 3.1 million per patient. Casgevy, which is the CRISPR-Cas9 product, is around 2.2 million. And this is definitely a huge burden for the healthcare system in the USA and everywhere. This definitely cannot be affordable for developing countries, especially for places that there are a lot of patients. And we need to continue our efforts to make these genetic therapies, which seems to be a very hopeful future, to be available for as many patients as we can. So what are the future challenges? Base editing definitely may be one of the new gene techniques that will solve some of the problems of gene addition and gene editing based on the non-homologous joining. We need to find some ways for improved conditioning, because as you can see, most of the toxicities are related to the conditioning regimen used. And hopefully in the future, we might find some way for in vivo delivery and no need for in vitro manufacturing of the product. So you can see that there has been significant advances in gene therapy for hemoglobinopathies. There's always a concern for the long-term effects and the risk for carcinogenesis. The gene editing seems to be a very promising field for a new therapeutic intervention in many diseases, not only for hemoglobinopathies. The cost remains, of course, the biggest obstacle for a wide use of this therapy, but for sure the future is brighter. And I would like to close here and thank also my colleagues at the clinic. Thank you very much for your attention. And we're glad to discuss any questions that you may have.
Thank you very much, Professor Kattamis, for this exhaustive presentation and very, very clear. So as you said, we have 15 minutes for questions or comment, if any. Just for you to know, you can both unmute yourself and take the floor, or raising your hand, or writing the question in the chat. I know you could take some moments for formulating them, or I also like to reverse the rule. And sometimes I say to the speaker that if you have some questions to share with the audience, as the audience are healthcare professional as you also could try to raise questions. So we'll see if anything pop up for taking them.
I don't know if somebody from UK is on the meeting, because I forgot to mention that UK was one of the first countries to approve these therapies. And I was wondering if they have any experience with these therapies after their commercialization in their country. But we may not have somebody from UK, so.
Yes, I don't know. I will just wait a few seconds because sometimes it takes some moments for raising. If not,
Maria.
means that everything. Yes, we have one hand raised. Please take the floor.
Okay. Hi, sorry, I'm on the move. This is Carsten from Cyprus. Very good presentation. Thank you so much. Hi, yeah, hello again. So very good, very updated. I was really happy. So I was wondering now, because you've been reading up on all of these approaches, and actually the two things you mentioned going out. Of course, the one being myeloablation or conditioning that is milder, and going in vivo. You as a clinician, because I'm a lab rat. So how do you see these things? So, for example, the approaches that are being used at the moment as CD117 antibody-based targeted killing of hematopoietic stem instead of in indiscriminate myeloablation as the one approach, which presumably would have much lower toxicity. And then, of course, the different approaches to go in vivo. The one where Lila Evangelia Nike is involved with CD46 targeting, with adenoviral vectors, but then also some companies that seem to be producing very specific synthetic nanoparticles for targeting. So this is very exciting. Maybe do this more critically, because you'll be at the sharp end when those become to be applied in patients.
Your points are very well taken, and this is the way to go. And yeah, the thing is that most of the as you know, most of the data we have so far are on preclinical basis, but that's the way things are going. Just to remind you that nowadays there is a lot of discussion about haploidentical bone marrow transplantation using a low conditioning regimen for sickle cell disease, which seems to be fairly good in inducing a long-lasting chimera that is actually can render the patients without any significant problems with sickle cell disease. So I think that's the way to go.
Another option. Okay, yeah. Okay.
Thank you. So then I see another question in the chat. What is a basic setup required to deliver gene therapy?
Well, so this is a very difficult question to answer, because you need to have a very strong lab that know how to work on CRISPR-Cas9 or some other gene editing techniques, or to be able to have the vector available if you like to do your own studies. At this point, as a product is commercialized, I think that's the only way to do is to have. I know for sure that the companies, in order to provide the use of their product, they need to have an established bone marrow transplantation unit, which has been accredited with some type of accreditation for gene therapy or for JC, for transplantation by an international agency. So these are the basic setup that you need, okay? And of course, the budget to cover the expenses.
Thank you. So I will announce next Thursday webinar, so next possibility to have a key international speaker sharing with us the cutting-edge advances in rare hematological disease. So we wait to see other last question arrive. So on the 1st of February at 5, we will have Domenico Girelli, Professor Domenico Girelli, talking about diagnosis and clinical management of rare forms of hemochromatosis. And so I hope to see you there. And I see no more questions. So, no, yes, sorry, there are hands raised. So Salvador and then Kennedy.
Okay. Thank you, Professor Kattamis, for your great talk. I'm Salvador from Spain. So, as you know, very good results were presented in us about haploidentical transplantation, as you said before. So we should start thinking about this platform with cyclophosphamide post in adults with sickle cell disease. So if we implement this, which do you think would be the role of each therapy in the future?
You mean if we implement this instead of gene therapy, correct?
Haplotransplant in adults. Which patients would go for transplantation and who for gene therapy?
Well, for haploidentical transplantation in older patients, although the results are very good, they are still not as good as gene therapy. And the main problem is that if it does not, well, the problem is with haploidentical transplantation, you may end up having significant graft-versus-host disease, which can be much worse than your initial problem. So at this point, although this is something that can be used, I don't think it's so safe and efficacious as gene therapy at this point, at least.
That it's true, but the cost is 5
Definitely.
times lower. So we should
That's
take
true.
this into account as well.
Yes, this is very, very good thoughts, and there's no good answer for that, because we need to think as scientists, we need to think as doctors, but also we need to think as the keepers of the health system. So it depends in which position you are in order to balance your decision. But I agree with you, this is a very delicate subject, and it's not a clear answer. It's not right or wrong.
Okay, thank you.
That's why we need committees to set up some rules and some guidelines in order not to have each one of us on the position to make this type of decisions.
Thank you.
Thank you very much. So I can pass the floor. Yes, please.
Kennedy, I think.
Yes.
Yes. Oh, sorry. My camera my connection is not good, so I can't do the camera.
Okay.
Sorry. Just two questions combined into one. So for the patients who are poorly responding or failing, or the response is not optimal, especially the adult patients you pointed out, what were the main reasons for the treatment failure? And then in this particular set of patients, now that you have some data, do you pick up any discernible changes in their metabolome or proteome? Thank you.
Okay, the second question is fairly easy. These studies are still ongoing, so I don't have any data to present. There are no public data available, so I cannot give you any results on that. And the first case, why there was a difference in efficacy, some of the cases did not have a good vector number, and one of the case for XSL, and also the same happened with the XSL, the transfused cells did not, were not, the percentage of edited cell was lower than the others, than the other patient. So that's the main reason of not responding so well. But the thing is that even the response was not, it was not 0 or 100%. For this patient, there was an improvement, but there was not as good as to be transfusion-free or VOEs completely free. But there was an improvement even for the patient that did not reach the primary endpoints. And there's one on the chat. Are there studies comparing haplo and gene? That's a very good question. No, there are not studies comparing haplo versus gene, and I don't think that will ever be possible to do this type of study without, because there's not going to be any funding for that. So that would be great, but I don't think we'll be able to.
Thank you very much, and we arrived at 6 p.m. So thank you very much, Professor Kattamis, for having shared all this expertise with us, and for the audience for the interaction. So maybe see you the 1st of February, and have a nice evening. Thank you very much.
Thank you for the hospitality. Take care, everybody. Bye-bye.
Thank you so
Bye.
much. Bye. Bye.
Thank you.